Welcome to our webinar, Innovative Workflows in Drug Discovery, Cell-Based Assays, and Next-Generation Sequencing with the I.DOT. Thank you for being here today. My name is Neela Lay, and I am the Marketing Manager here at Dispendix. We have registrants from all over the world, and I'm so proud to be moderating this webinar. On today's agenda, we will hear from an exciting panel of experts in cell-based assays and NGS. Please feel free to use the Q&A feature throughout our program, and we will address your questions at the end. Let's get started. Before we begin and bring our esteemed speakers to the stage, I'd like to take a moment to introduce the groundbreaking technology that is at the heart of our discussion today, the I.DOT. For those of you who are unfamiliar, the I.DOT is an automation-friendly, non-contact dispenser that's revolutionizing the way we handle liquids in the lab. Let me highlight some key features. The I.DOT can transfer volumes as small as 2.55 nanoliters up to 30 microliters. It's compatible with 96, 384, and 1536 well plates, offering incredible flexibility. It can handle a wide range of liquids as well, from methanol to 65% glycerol. I think the best way to show you how the I.DOT works is by watching a short explainer video whose voiceover you may recognize. Give me one moment while I pull it up, and please enjoy.
The I.DOT is an automation-friendly, non-contact dispenser designed to transfer nanoliter and microliter scale volumes to 96, 384, and 1536 well plates. 96 well I.DOT source plates can dispense any liquid with viscosity from methanol to 65% glycerol. One channel of the dispense head and one well of the source plate combine to form a dispensing channel. The dispense head modulates air pressure, which can be precisely adjusted to generate droplets from 3 to 50 nanoliters at a nanoliter resolution of 0.1. Each channel generates 100 droplets per second into the target well. A 96 well plate can be filled with 100 nanoliters per well in 7 seconds. To conserve valuable reagents and samples, the I.DOT requires less than 1 microliter of dead volume. To provide users with the highest degree of process safety, the I.DOT counts the number of droplets dispensed with miniaturized light barriers mounted underneath the source plate. If source wells run out of liquid during dispensing, the I.DOT can backfill the missed droplets, preventing user error. The I.DOT enables fast, precise, and traceable liquid handling, enabling the most efficient and reproducible genomics, drug discovery, and synthetic biology workflows.
With this introduction, I hope everyone now has a basic understanding of the I.DOT. This technology will be central to the innovative workflows our speakers will be discussing today. If you have any questions about the I.DOT, still, I hope that our speakers will answer some of them through their presentations. And if not, feel free to ask them using the questions feature. And now, without further ado, let's welcome our distinguished speakers to the stage. The first speakers are Andrea Krahn and Wolfgang Reintsch. Andrea was born in Mexico City and later raised in the small town of Niagara-on-the-Lake. She attended Brock University, where she received her Bachelor of Science in Neuroscience. She then worked as a chemical analyst at the Canada Centre for Inland Waters before moving to Montreal to pursue a master's in neuroscience at McGill. She is now a research assistant at the Neuro's Early Drug Discovery Unit, or EDDU for short, working on automation of assays in iPSC-derived neurons for high-content screening. Wolfgang has been a part of the EDDU since 2017, where he manages the assay automation and high-content screening platform and oversees the discovery of assays and screening group. Before joining the platform, over the course of 10 years, he worked in several industrial laboratories in Montreal with a focus on automation, high-content microscopy, and image analysis. He managed a light microscopy and molecular biology platform in the Department of Pharmacology at McGill University. Wolfgang received his PhD at the Max Planck Institute for Developmental Biology in Germany. His work there and later as a postdoc at McGill University circled around the cellular mechanisms that shape the developing embryo. During that time, Wolfgang started to develop microscopy tools and automated approaches for his research, and he has maintained a strong interest in this area ever since. Andrea and Wolfgang, please take it away.
Thank you for having us today. So as Neela mentioned, Wolfgang and I are part of the automation and screening platform at the Early Drug Discovery Unit. We are located at the Montreal Neurological Institute, which is part of McGill University. And today we're going to be talking to you about some of the ways in which we've incorporated the I.DOT into our platform to test for compounds in iPSC-derived neuronal cell models. So before we get into all the wonderful things that the I.DOT can do, I would like to give a brief introduction on who we are and what we do. So here at the Neuro, we work in collaboration with CBIG, which stands for Clinical Biological Imaging and Genetic Repository. They collect samples from healthy controls and patients with various neurodevelopmental and neurodegenerative diseases, such as autism, ALS, and Parkinson's disease. We can then take these PBMCs from blood collected or fibroblasts from skin biopsies and reprogram them into induced pluripotent stem cells, or iPSC for short. We can then use CRISPR-Cas9 genome editing to correct disease-associated mutations in a patient line in order to generate an isogenic control. We can also insert mutations or knock out specific target genes. And currently, we have a catalog of over 200 iPSC lines that we've generated. Since we're interested in diseases of the brain, we can then take these stem cells and differentiate them into neurons, like dopaminergic, cortical, or motor neurons, as well as glial cells, like astrocytes, oligodendrocytes, and microglia. And we can take these cells in either 2D or 3D cultures to develop assays and screen for compounds. I would also like to mention that the Neuro is an open science institute, so all of our iPSC lines are freely accessible for experimental research. So if you're interested in learning more about that, we will have a link to our data portal at the end of the presentation. So this is what a typical workflow would look like for us. We use a washer dispenser to coat plates, seed cells, and do media changes. We can then use a pinning tool to transfer libraries of compounds. And once we've selected candidates from thousands of compounds being tested, we can then further these compounds' effects using the I.DOT. And today we're going to be showing you 3 ways in which we use the I.DOT for this. We can then use a washer to fix and stain cells, and then further use various automated microscopes to image and analyze our plates. One of the diseases we're interested in is Parkinson's disease, with one of its hallmarks being the accumulation of alpha-synuclein fibrils inside the cells. So alpha-synuclein is a small 14 kilodalton protein that typically exists as a monomer, but can misfold and aggregate, causing fibrils that will accumulate into the cell. These aggregates are then thought to be able to spread from cell to cell in a prion-like manner, ultimately causing cell death, particularly the dopaminergic neurons in the substantia nigra of the brain. And the 3 examples I will be showing you today were actually done in dopaminergic neuronal cells. So one way in which we can study the aggregation of these alpha-synuclein fibrils is by using recombinant fluorescently labeled fibrils that we create in-house. And then look at whether certain compounds can decrease the total fibril load inside of the cells. And here's an example on how the I.DOT can simplify a process like this for us. So as you can see on the left, we can add various compounds into our source plate, and then the I.DOT will then go and dispense them in a dose response manner, as you can see on the right, into our target plate. We also have DMSO controls in green, as well as wells that contain only media to normalize for the volume in each well. We incubate compounds for an hour before adding our fluorescently labeled fibrils, and then we can image them the day after. So as you can see, these cells readily take up these fibrils and they accumulate in the perinuclear area over time. But if you add certain compounds, you can see there's a decrease in fluorescence around the nucleus. We use automated software to quantify the fluorescence intensity in the perinuclear area by using Hirsch as our counterstain and creating a mask around that. And on the right, you can appreciate what our typical dose response would look like. So with increasing concentrations of the compound, there's a decrease in fluorescence intensity. And we can also extract features, nuclear features, such as cell count, nuclear area, and nuclear intensity to monitor for cell health. So now I'm going to pass it over to Wolfgang to give you a couple more examples on the I.DOT.
In this slide, I will present a project that is led by Martin Berrier and Carol Chen at the EDDU. Their overall arching theme is the morphological phenotyping of dopaminergic neurons. More specifically, right now they are developing methods to distinguish neuronal cultures from Parkinson patients from healthy individuals. Being able to do this then allows to screen for small molecules that allow to revert the phenotype of a Parkinson patient line towards a more healthy phenotype. And this could help to interpret the morphological pathways that are involved and maybe lead then to a therapeutical indications. In general, looking at the morphology of these neurons in culture, they're relatively similar. So Parkinson patients and healthy lines, they look very simple when looked through the microscope, and even by conventional image analysis methods, that's meaning extracting specific features like, for example, cell length, branching points, cell size, they are pretty much indistinguishable from each other. Martin and Carol decided to use a different approach that was inspired by cell painting. Cell painting is a method that was developed originally in Ann Carpenter's lab at the Broad Institute. This method uses multiple dyes against different cell compartments to stain the cells overall and then extract unbiased and every feature possible, so hundreds or thousands of features from these images, to be able to then create sort of a fingerprint of these cell types that allows to distinguish different cell types and phenotypes from each other. One issue with this approach is that it's unbiased, so it's relatively sensitive. Things like, for example, or mainly plate effects, which means, in other words, that the position of a culture in the well might already influence its morphology or its reaction to the compounds added. So to avoid this, we use the I.DOT to add compounds at relatively random locations within 96 well plate or in the culture plate. And this is shown here. So the stock solutions are simply loaded on the left in the source plate and then distributed randomly at different doses into the target plate or the cell culture plate. And this was done usually in several replica plates at a time in one experiment. So doing this by hand would have been very laborious and error prone. With the I.DOT, it took a few minutes per plate at max. So on the right, I showed there are some of the results. So in this case, the cells after 2 weeks of culture of differentiation were stained with 5 different dyes against different cellular compartments. Then several hundred features were extracted from each image. They were analyzed by machine learning approaches, and then the 50 most relevant features were visualized using principal component analysis. Showing in this graph, so the red circle is the Parkinson cell line, the square, the control cell line, well separated, and then there's an example of one compound at 3 different concentrations. At the small diamond next to red circle is the lowest concentration, meaning the morphology is relatively similar to the Parkinson line, while the higher doses, the morphology shifts more towards the control line. So this is our last example. The overarching theme here is that looking for synergistic effects on neural network density. So in this case, neurons were differentiated for 4 weeks, and during the last 2 weeks treated with compounds in different combinations to see if these compounds could improve neural health judged by the density of the neural network. Adding combinations of different compounds in the same well would have been quite complicated, so the I.DOT solved this with ease. Again, on the source plate, we just load the stock solutions of each compound and the DMSO control, and then it distributes either in parallel or in sequence the different compounds into the different wells. So each, as you can see then in this very colorful cell plate depiction. So again, this would have been, I think, almost impossible to do manually. Plus, we could easily do 6 replica plates per experiment, and it took a few minutes per plate, as I mentioned. Results on the right. So we stained the cells at the endpoint after 4 weeks with pan-neuronal markers like neurofilament heavy chain or beta tubulin, and then did a relatively simple analysis in this case, which is to threshold against the staining. So essentially measure the total area of the neuroid network on each image, then normalize it against the untreated control. And in the graph, you can see then the results. So in a most simple way, so what you can see is that there's a small but significant difference in this case between a combination of 3 compounds, A, B, C, and the untreated control. And this is true for both markers, both neuronal markers. So indicating that this combination of compounds boosts in some way the neural connectivity and the robustness of the neural network. I hope by showing you these 3 examples, you get a good idea of how we use the I.DOT in the lab. To just repeat what Andrea said, we have a lot of, well, we use instruments all along our workflow to improve our throughput, to decrease, and also to decrease variability and increase the robustness of our assays. And in terms of compound addition, the I.DOT had really been checking all the marks, and not the least by allowing us to do experimental setups that we wouldn't have even thought about before having it. So I want to thank everybody at the EDDU and our boss, Tom Durkin. And thank you for listening. If you're interested in the EDDU or the automation platform or the CBIC and the iPSC repository, please follow the link, and thank you for your attention.
Thanks so much, Andrea and Wolfgang. Such an insightful presentation. Our next speaker is Kevin Mohammed. Kevin Mohammed is an instructor at the Mount Sinai School of Medicine in New York, with a distinguished background in molecular biology and virology. He earned his PhD from the Rockefeller University, specializing in HIV molecular biology, and graduated summa cum laude with a Bachelor of Science in Biology from Morehouse College. With over a decade of research experience, including key roles at the Aaron Diamond AIDS Research Center and Girelet Incorporated, Kevin has made significant contributions to the understanding of HIV-1. He now works in the Guccione Lab, which focuses on the function of methyltransferases in development and disease. The lab uses a combination of approaches to study the function of PRMTs and PRDMs. Kevin, the floor is yours.
Thank you so much, Neela. As Neela mentioned, I work at the Mount Sinai School of Medicine in New York City, and specifically in the Center for Oncogenomic and Innovative Therapeutics. This is a center that was opened maybe about 2 years ago, and it's an offshoot of the Guccione Lab. And the goal of this center is to provide a platform for precision oncology drug discovery, specifically by using multi-omics tools in order to understand cell fate transitions and to develop innovative therapeutics for cancer. So this is a little snippets of the kind of research that our center does. So we're involved in drug development. Recently, we published a paper about a small molecule multi-kinase inhibitor that we discovered in a drug screen from an in-house manufactured library that we screened through hepatocarcinoma organoids. And also to highlight the interest that we have in more basic mechanisms of cancer biology, We also have a paper in press right now, after 3 years of work, highlighting the transition of cellular states and how it applies to the escape of cancer cells in colorectal cancer from therapeutic intervention. So our lab is based in Mount Sinai, and it's strictly has been an academic setting. We have very little instrumentation or expertise in doing high throughput kinds of methods. We invested in the I.DOT perhaps 2 years ago in order to bridge that gap between our medium to low throughput methodology and high throughput methods. And I could say unequivocally that the I.DOT has been an extreme boon to our research efforts. For instance, we use it all the time to load qPCR plates, whereas it would take me personally, it would take me an hour to load 384 wells at least. The I.DOT is capable of loading 384 wells in 15 minutes. So everyone is extraordinarily happy with the instruments with regard to this. We normally use this also to normalize nucleic acids when I do library preparations. We could do 96 DNA or RNA samples in minutes. We also use it routinely to generate IC50, IC80 drug curves. And this is done in 5 minutes. And also, while I'm going to talk to you today about the efforts that we're trying to use in order to incorporate the I.DOT into our NGS workflows. So a little bit of background on the system that we've been using. Well, one of the systems that we've been using. Colorectal organoids are these three-dimensional structures that you could grow on Matrigel, and they are derived from these stem cells that you can see in the center panel in green, which are capable of self-organizing into these three-dimensional structures, which mimic the adult intestinal niche, as you can see on the right-hand side. You get crypt domains with the stem cells at the bottom, and then there are the villi domains, which approximates what the villi look like. So what is very cool about this organoid system is that we can artificially introduce mutations that are naturally found in colorectal cancer progression. So we could see the organoids at the bottom level, the wild type make these nice, beautiful structures. And it's very interesting to see as each subsequent mutation is introduced that the phenotypic structure of the organoids actually changes along the spectrum. As I mentioned, these organoids, if you look at the right-hand panel, specifically at the wild type in blue, most of the cells exist in the enterocytes as enterocytes, and also a sprinkling of these other naturally occurring cells that you find in the epithelium of the intestine. And you can see that in the precancerous cell lines, the A cell lines, that there's a deregulation of development. There are few enterocytes and upregulation of these stem cells. And there's also this pattern that you can see in the AKSP model, which represents the malignant form of the disease. And I just wanted to highlight these stem cells here. And these are important because they are believed to be the agents responsible for relapse in patients who have undergone therapeutic intervention, since they are capable of regenerating the tumors either at the original site or at distal sites and metastases. But the importance of these stem cells have been called into question lately, because if these stem cells are ablated, these patients can still develop relapse. So there's clearly some other thing going on, some other cell population that might be present that's responsible for cancer relapse. So the first thing that our group did was we analyzed these different colorectal cancer organoid systems with single-cell RNA-seq. And you could see that in this UMAP profile, there's a very small population of cells that's upregulated across the disease intensity. And we found within this subpopulation of cells, there's a specific set of genes that are upregulated. And these cells have some characteristics of stem cells, but they also express these other genes, which are expressed uniquely in fetal tissue. And they also express other genes that are mostly expressed in anterior sections of the gastrointestinal tract, including genes expressed in the stomach, showing that there's a retrograde evolution of these cells back to an oncofetal state. This collection of genes is something that we dubbed the oncofetal signature. So very interestingly, when we treat the cancerous organoid system with FOLFIRI, which is one of the chemotherapeutic agents against colorectal cancer, and they're treated for 3 days, the cells that do survive this drug treatment actually are enriched for this signature of oncofetal genes, which may indicate that these cells may play a role in the relapse of cancer. So a lot of these experiments we did for this paper were done on either 6-well plates, 24 or 12-well plates. And we're interested in miniaturizing things because we're interested in doing some drug screens that could perhaps modulate these cell states decisions. And of course, it's untenable to do these kinds of experiments with these large plates. So we wanted, first of all, to miniaturize things onto a 3-4-well plates. And we wanted to do some proof of principle experiments to show that, yes, we can grow organoids on this smaller plate. And if we could actually treat these organoids with drugs, and if we could detect this oncofetal signature. And this is where the I.DOT really played a part, because the volumes in these 3-4-well plates are 50 microliters, and it's a very little small amount of drug that needs to be added to every well. And as a bonus, we'll also save a lot of money on media, which is very expensive to grow these organoids, matrigel, fewer drugs will be used, and also less RNA extraction reagents. So this is the approach that I use in order to miniaturize things, a 3-pronged approach. First of all, miniaturization of the tissue culture system, and then to move away from what we traditionally use, which is column-based methods to extract the RNA, to a plate-based method and magnetic beads. And then also a different library prep technique, which promises to deliver less labor and lower cost, called BRIPSeq. So in the first set of experiments in miniaturization, we tasked the I.DOT with dispensing drugs onto the 3-4 plates with our organoids. And we did this dose response curve, starting from 100 micromolar all the way down to 1.7 nanomolar. We're constrained at the upper limits by the amount of drug that we could dispense. So we could only do 0.5% DMSO. So at a maximum, we have to dispense 250 nanoliters. So that's well within the range of the I.DOT. It does it beautifully. And then at the lower range of the physical limitations of what the I.DOT can dispense, at least in the source plate that we've been using. We only had to do 4 different drug concentrations, and this was able to incorporate the entire range of drug concentrations that we needed. So we are very happy to see that we got very beautiful dose response curves in response to FOLFIRI. These look exactly like the kinds of dose response curves that we get in higher, larger tissue culture plates. We use the new technique of isolating RNA with magnetic beads, and we got very nice yields of RNA from these 3-4-well plates, 800 nanograms, which is more than enough for the library prep process. And interestingly, the RNA profile looks very similar to the FOLFIRI, which makes us very confident that we're getting exactly what we put in. So I've told you about the efforts to miniaturize and RNA extractions, and I'll talk a little bit about the efforts to make the library prep process more high throughput and cheaper, the BRIPSeq, as you see on your right-hand side. So bulk RNA barcoding seq, or BRIPSeq, is a technique that was published in 2019 by this Swiss group. I'll quickly go through the protocol. The BRIPSeq is dependent on this oligo, which incorporates 4 different elements. So it has the poly-DT, which allows annealing to the mRNA transcripts. It has a unique molecular index, which identifies the specific mRNA that it captures, and has a sample-specific barcode. Finally, it has a handle at the end, which allows for PCR amplification of the specifically captured mRNA molecules. Secondly, the first strand cDNA synthesis is done. Second strand is made, and then these are cleaned up and then subjected to tagmentation with TN5 transposase, which cleaves the double-stranded cDNA, and then the specific 3' end, which has the sample-specific barcode, that little fragment of the gene, and the TN5 adapter is amplified by PCR, and then this goes on to the sequencing of paired end chemistry. Read 1 gives you the cDNA that is captured, and then read 2 reads sample-specific barcode and also the UMI. So the pros of this library prep process is you have, first of all, early barcoding. So everything is barcoded at the very get-go. So this reduces any chance of cross-contamination at subsequent steps. There are fewer steps, which equates to less labor, less chance of error. Fewer reagents are used, which I'll show you in a subsequent slide, which translates into less cost. And then also, because you're only sequencing the 3' end of the gene, these sequencing reads are not spread throughout the entire gene body. So you need fewer sequencing reads in order to get the same information using this library prep technique. Cons, however, is since we're doing 3' end sequencing, there's no information on RNA isoforms or alternative splicing. And also there's no information on any kind of mutations, such as insertions or deletions. So this highlights the beauty of this library prep process in this theoretical experiment in which you have 96 samples being processed. All 96 samples are loaded onto a 96-well plate, and first strand synthesis is done with 96 unique barcodes. And then, since everything is already barcoded, all the samples can be pooled into a single tube, cleaned up, and then from there on out, all 96 samples are processed in a single tube. So you can see how the labor costs and the pain and suffering of doing library prep is dramatically reduced using this technique. And this is in contrast to conventional RNA-seq, where you have to do the exact same thing to all 96 samples through every single step of the library prep process. I like this slide. This is a cost analysis per sample. So for conventional RNA-seq, that cost remains constant no matter how many samples you have. In our hands, we use New England Biolab's kits, which costs approximately $36 per sample. And the BRIB-seq, even though it's very expensive, if you do just a few samples, because the TN5 is a very expensive enzyme, the break-even cost occurs at just 4 samples of BRIB-seq. More samples are put into the project, and the sample numbers are on the x-axis, that we approach an asymptote of approximately $6 per sample during this library prep process. And this is actually a cost that we want to reduce even further by making our own in-house TN5, which should approximately, we hope, half the cost of this library prep per sample. Relatedly, this is the cost of a total project. If you have 96 mRNA, conventional mRNA-seq libraries, cost is approximately $3,500, whereas 96 BRIB-seq samples cost just a fraction of that cost. All that's good and proper, but we wanted to see if the BRIB-seq library prep process can identify this oncofetal signature in our miniaturized system. So can we treat these cells on this 3D4 well plates with FOLFIRI and get the same results we get from a larger organoid system with conventional RNA-seq? So we are constrained. Obviously, we're going to do experiments on 3D4 wells, so we're constrained by the number of cells that we could test. And also, we are going to test the BRIB-seq with just 25 nanograms of input. We choose this because this is the drug dose at which we see this upregulation of oncofetal signature. So this is an overall flowchart of what the initial pilot experiment was in order to assess if the miniaturization is capable of detecting this oncofetal signature. We had FOLFIRI loaded onto the I.DOT, and we had it dispense that same drug curve that I showed you previously, 250 nanoliters down to 28 nanoliters. We left the drugs on the plate for a couple of days. We extracted RNA using magnetic beads, and the RNA was eluted directly onto the source plate, which was loaded onto the I.DOT again. And then we asked the I.DOT to dispense 1 microliter of each RNA sample onto a 96-well qubit assay. A few minutes later, we have the RNA concentrations. We feed this back into the I.DOT in order to ask it to dispense 25 nanograms of RNA in a normalized fashion onto a 96-well plate. Oh, and by the way, this pilot test was done with 66 samples at once. So all 66 samples were loaded onto the 96-well plate for BRIB-seq protocol. They were pooled after first strand synthesis, and then loaded onto our NextSeq 2000 using a P1 flow cell, which gives us approximately 140 million reads. And we sequence a very shallow at 1.5 million reads, which coincidentally is just very cheap at just $9 per sample. So we see in our analysis that there's a beautiful dose response to FOLFIRI concentration compared to DMSO control. And we see that there's a lot of upregulated and downregulated genes as full ferry treatment is increased. And we were very, very happy and very relieved to see that when we analyzed the upregulated genes, that a lot of the genes that are found in the oncopetal signature were in fact discovered in this miniaturized process. So this ultimately will be a typical experiment once we get everything nailed down. I won't go through the initial process on the left-hand side, but the I.DOT in the middle there will be used to dispense the RNA. And once that is done, the rest of the RNA will be dispensed onto 96 well plates or other, and it's frozen and archived for backup. And then each of these 496 well plates, which represent the entire 384 wells, can go through the BRIP-seq process, pooled into 4 tubes, and then sequenced. So this is the idea of what's the ultimate screen that we want to do. So that's the end of my talk. So I hope I was able to convey the kinds of things that we're doing with the I.DOT. We are extraordinarily happy with how it's contributed to our workflow in the lab, and we look forward to using it in many, many more applications. And of course, I have to thank everyone in our lab, especially Ernesto Guccione, who's the head. And of course, I have to acknowledge Slim and Megan, who have done 99% of this work in the organoids and written this paper up. And of course, I want to thank the people at Dispendix, Maya and Robert. Maya was at the very get-go helping us to purchase the instruments and to get everything up and running. And then Robert Fries was able to get experiments up and running, and he's always available via email to help us out to troubleshoot. And he's been an extremely helpful resource at Dispendix. Thank you so much. Thank you for listening to me, and I'll give it back to Neela.
Thanks so much, Kevin, for your insight, especially our distinguished speakers. Thank you so much, Andrea, Wolfgang, and Kevin, again, for your time today and for sharing your expertise with the scientific community. No matter where folks are in the world, we at Dispendix thoroughly enjoy connecting with the scientific community, and we'd love to hear your feedback. Have a great rest of your day, everyone, and we look forward to hosting the next webinar soon. Thanks so much again.